Okay, so the broad topic for today's discussion will be the idea of genome instability and how genome instability can lead to cancer, basically. Because my understanding is that the theme of this program overall is the sort of biology of cancer. And so I like to think that where we are, what we'll be discussing today is actually starting at the very beginning of the process, which is sort of the very early events that occur in cells that eventually lead, sometimes years, decades later, to the onset of tumors. Okay? And so the topics for today then are going to be just a general discussion of what genome instability is, and importantly, what causes it, and then some indication that it does in fact eventually lead to cancer. We're then going to talk about the concept of checkpoint control of the cell cycle. I understand you heard from Andrew Murray already a little bit about how checkpoints sort of operate in a philosophical sense. And we will be focusing on how checkpoint systems actually guard against the onset of genome instability. And then finally, at the end, we'll be looking at some primary data, a couple of papers that have come out very recently, which describe ways that cells can actually physically detect the presence of damage, okay? Because that's obviously very important if cells want to mount a response to that damage so that damage does not lead to mutation and eventually to cancer. So what is genome instability? Well, the best way to describe it is to actually show you a picture of it, and this is what it looks like. The top panel is a karyotype, where you're looking at normal chromosomes from a normal cell, from a normal person. And as you can see, each chromosome is represented by two copies, except for the sex chromosomes, one each, X and Y. And this is essentially more or less a stable genome, at least at this gross sort of morphological level. By contrast, I think you can appreciate the dramatic differences in gross chromosomal morphology and content between a normal cell and the panel that's shown at the bottom, which is a karyotype derived from more or less typical cancer cell. And as you can see, this karyotype shows very clearly the hallmarks of an unstable genome. You have multiple copies of some chromosomes. Other chromosomes are completely missing. And importantly, you have a whole sort of collection of oddballs here, which are really the result of translocations between normal chromosomes to create aberrant chromosomes, if you will. And so this is what happens over time to create this is what genome instability does at the chromosomal level, and the result of that is that you can get cancer, that cancer cells can form. And so the types of mutations that basically can occur on DNA that can eventually lead to cancer are a broad number of different types. And they're characterized generally by either insertions or deletions, where exogenous sequences are added to a chromosome or endogenous sequences are removed from a chromosome to create a deletion. You can have simple point mutations, which simply change the nature of one base, for example, C to A. Translocations are very common in cancer cells. That's where big pieces of chromosomes are moved from one onto another. And related to that are intact chromosome fusions, where whole chromosomes are fused together. So those are the types of mutations that can occur when genomes become unstable. And how this relates to cancer is kind of summarized in this slide here, which goes over the basic theory behind the requirement for genome instability and the eventual onset of cancer. And the theory says that genome instability occurs when the mechanisms that guard against mutation, and there are a number of different types of mechanisms, and we'll talk about them in a moment, when they collapse or fail. Okay, so this is really sort of step one in the whole process. Interestingly though, this in and of itself is not a horrible thing to the cell. You can lose all ability to monitor the genome, and that's okay as long as nothing bad happens. Now the bad part is, unfortunately, it's very rare when nothing bad happens. And what does occur in cells that have lost the capacity to protect against mutation is that obviously mutations will arise and occur at high frequency. And then occasionally a a cell can pick up a mutation in a key cellular growth control gene. And this is when things start getting really bad, okay? Because the mutation, or the ability of cells to tolerate a high mutation rate, only becomes problematic when those mutations begin to affect the behavior of proteins that are necessary to regulate how the cells divide, okay? So picking up mutations in key growth control genes can result in misexpression of the particular gene or loss of the particular gene. And this then can confer a selective growth advantage to that cell containing that particular mutation. And this is a really important concept here. We'll go over it more so in a moment. And the other thing that's important to appreciate about the general theory is that it's not a single mutation generally that causes cancer. Rather, it's the accumulation of mutations in multiple critical growth control genes. And this can happen by activating pro-growth genes inappropriately, as well as, and perhaps more commonly, by inactivating genes that exist to limit growth, and those are otherwise known as tumor suppressor genes. Okay, and so that theory then can be sort of summarized in this little flow chart here, where the first thing that happens in cells is a failure in cell cycle checkpoints, and cell cycle checkpoint is just another term that can broadly be used to describe how cells prevent genome instability. Okay. So the failure of the checkpoint leads to genome instability. That eventually leads to a mutation in a key growth control gene. That then confers a selective advantage onto that cell, which allows it to expand more rapidly relative to the rest of the population, thus giving it a better chance of picking up yet a second mutation. Okay? And you go through this process 6, 7, 8 times, and eventually you can get a tumor forming, okay? And so a key component of this theory is this concept then of clonal selection and expansion. Okay, this is almost necessary in order for us to imagine how human beings can develop cancer within the lifetime of a normal person, 70 years, for example. And the theory says that, again, normal cells are doing their thing. You generate a mutation. That confers this growth advantage, so the cell begins to, in a modest way, take over the population. It's by no means a full-blown tumor yet, but it does have a growth advantage over its neighbors, and that's the critical thing. That increases the chance that a cell of this genotype will pick up a second hit, again conferring an even more rigorous growth advantage now, expanding this pool of pre-cancer cells, if you will, precursor cells, and again increasing the chance that a cell of that genotype is going to then pick up the third hit, okay? And so on we go until eventually the critical combination of mutations has been acquired within a single cell or a single genome that allows that cell to then divide in really a very uncontrolled manner and forming a tumor. So that's the theory, and then what then is the evidence that a failure in checkpoint control is really a critical or sort of the initiating event in getting the ball rolling here? Well, that evidence is very persuasive, and it comes largely from essentially looking at human genetic diseases that confer a predisposition to cancer, okay? And I've listed a couple of them here. So mutations in an enzyme that's known to repair DNA damage, called MASH2, the function of this enzyme is actually to repair mismatches in DNA when DNA replication makes a mistake. And people who have mutations in this gene have a much higher chance of eventually developing colon cancer than do people who don't. Two more prominent examples of how having a mutation in a checkpoint or a genome guardian gene, if you will, can predispose one to cancer comes in the example of BRCA1 and BRCA2, which are two genes that are known to predispose women to breast cancer as well as ovarian cancer. I'll talk a little bit about more of these in a moment. And then another very good example of all of this comes in the form of a human disease, essentially, called ataxia telangiectasia, which fortunately is rather rare. and is caused by mutation of a gene called ATM, which really then predisposes people who have lost ATM function to a wide variety of tumors. And we'll discuss more about this in a moment. So this is just really the tip of the iceberg in terms of genetic evidence that says that if you knock out these systems that really function to maintain the stability of the genome, and that comes in the form of either actually directly repairing the DNA or allowing cells time to repair the DNA by promoting cell cycle arrest, for example, that really that knocking out or compromising this system is a very critical event in terms of getting the ball rolling towards eventually getting a cancer.
Has it been estimated how many years this process takes from, say, step 1 to step 2?
Yeah, so it can take 30, 40, 50 years, essentially, based on statistical analysis when one factors in the rate of mutation and then the accelerated rate of mutation in cells that have lost checkpoint control, for example. But given the size of the genome and the exact number of mutations that need to accumulate in the right order, that's important. I didn't emphasize that earlier, but not only do you have to get to 7 or 8 critical mutations, but very frequently in cancer, those mutations have to arise in a predetermined order. Then, that can take a very long time. But unfortunately, it can certainly happen, obviously, within a human lifetime. So the idea then is that the maintenance of genome stability is really very critical for preventing cancer. And again, I think a very sort of powerful example of that is shown here by just pure statistical analysis on the risk of women for developing either breast or ovarian cancer when they suffer mutations in this gene called BRCA1. Now, the exact biochemical function of BRCA1 is still really unclear. What is clear, though, it is certainly involved in some aspect of maintaining genome stability and preventing mutation, okay? And so normal women who have healthy, intact copies of BRCA1 throughout their lifetime run about a 10% chance of getting breast cancer and a 1.8% chance of eventually getting ovarian cancer. But if you look here at women who are born without wild-type copies of BRCA1, the numbers are very, very striking. It goes up to 82% as a chance for generating breast cancer and 54% for ovarian cancer. So not having this particular gene is really very critical in terms of the ability to hold off the eventual onset of breast cancer in women. Question? There is a screen certainly available, yeah. And because of just the overall striking effect that screening has revealed, very often women who show no signs of developing a breast cancer will, but have been screened and found to lack this particular gene, will actually undergo mastectomies in order to avoid the eventual onset of breast cancer. Okay, so this is a very strong predictor of that. Question? I actually don't know the numbers for that. I think there is a gene dosage effect, but I don't know for sure, so I'd like to hold off on saying yes or no to that. I believe there is, but I'm not sure. Yes, these are homozygous mutations. So this is a germline mutation in these women. Question?
About 10% of men get breast cancer. Have there been any studies with men?
Yes, there is a link between BRCA1 and breast cancer in men also. Yeah, it's not as pronounced as this, I don't believe, but there is a link between the two. So one interesting aspect of this whole situation really actually also is this particular gene is known to be expressed in every single cell type in our bodies in both men and women. And why its loss results in a particular sensitivity to breast and ovarian tissue to cancer is another very important but unanswered question in this general field. Okay, so I think then that these examples really of basic epidemiology and human genetics underscore the importance of maintaining strong checkpoints and the ability to maintain genome stability in order to prevent the eventual onset of cancer. And so now I want to shift a little bit into a discussion of how these genome stability, genome maintenance networks actually function at the molecular level. Okay. And so the first part of that discussion will just be a brief discussion of cell cycle checkpoints. Now I understand that Andrew Murray has already addressed this audience, and presumably he gave a discussion of the spindle checkpoint, which is a system that allows cells to monitor whether or not chromosomes have attached to the mitotic spindle. Okay, so cell cycle checkpoints, as Andrew probably mentioned, come in a number of different varieties, and of particular importance to today's discussion are those checkpoints that actually monitor whether or not DNA is damaged and react to the presence of damage. Okay, so that's different than what Andrew was discussing with the spindle checkpoint, where there the DNA is not necessarily damaged, it's just the chromosome is having trouble aligning on the mitotic spindle. And so the general idea is that cell cycle checkpoints monitor cell cycle progression. And when there are problems in chromosomes, for example, a lack of attachment to the spindle prior to entrance into mitosis, or the presence of DNA damage, the checkpoint then intervenes, it arrests the cell cycle, and it allows the cell time not only to do DNA repair, but also to stabilize any sensitive intermediates in metabolism that are present while the problem is being fixed. Okay, so that in general is the function of checkpoints. And another way of thinking about checkpoints then is the idea that they provide a feedback control on cell cycle progression. Okay, and so if things haven't occurred properly or normally, if the cell hasn't finished replicating its chromosomes, then there's a feedback control that says don't enter mitosis yet. And if the chromosomes are damaged, there's a feedback control that says let's halt progression through the cell cycle until we have repaired the damage. And so I just wanted to give some examples of early demonstrations of the idea of feedback control and how people began to first realize that these were really independent regulatory systems that functioned purely to monitor how cells were progressing through the cell cycle. And so a famous example of feedback control in mammalian cells comes from an experiment done by two guys named Rao and Johnson. Now, how many people here are familiar with the Rao and Johnson experiment? Is anyone? Yeah, this is one of the most famous experiments in cell biology and really set forward the idea that cells do have the ability to monitor where they are in the cell cycle. And the way this particular experiment was done is that two cell types, they're the same type of cell, but they're simply in different phases of the cell cycle. Okay, so cells from different phases of the cell cycle were fused so that now the two nuclei share a common cytoplasm. Okay, and so the ability to fuse cells is fairly technically trivial. Nonetheless, the question that Rao and Johnson wanted to ask then is within this heterokaryon, if you will, which of these two nuclei are going to, how are these two nuclei going to react to each other within the context of cell cycle progression? So in particular, if you take a cell that's in S phase and you fuse it to a cell that's in G2, will the cell that's in G2 enter mitosis on its schedule, or will it adjust its schedule to that of the S phase cell? Okay, and so the cells were fused, and then they were simply, the nuclei were monitored to see when they entered mitosis. And what Rao and Johnson found was despite the fact that the G2 cell had finished DNA replication and was ready to enter mitosis, upon fusion with a cell containing an S-phase nucleus, the G2 nucleus waited. It waited until the S-phase nucleus had also finished DNA replication, and then the two nuclei entered mitosis synchronously. Okay? So this very simple experiment says that cells have the capacity to recognize that there is an alteration in the cell cycle, and they have a means of delaying progression through the cell cycle sort of in reaction to that. Okay? And another interesting experiment was done by a guy named Art Pardee. Again, all of these types of experiments were done in the early 1970s. demonstrating again the idea of feedback control in mammalian cells, where S phase cells were treated with a drug called hydroxyurea. And hydroxyurea simply acts to block DNA replication. So if you take an S phase cell and you treat it with hydroxyurea, it will remain in S phase forever because it can't complete DNA replication. And what Pardee found is that if you co-treated those cells with caffeine, then you could advance this S phase arrested cell into mitosis inappropriately. Okay? And so what the experiment says, taken together, is that the reason that this cell did not enter mitosis was not because it couldn't. It had the capacity to enter mitosis. It's just that it knew the DNA replication had not yet finished, so it chose not to enter mitosis. When caffeine was added, that overrode that control mechanism that said, let's not enter mitosis because replication hasn't finished, and thus promoted entrance into mitosis under inappropriate conditions. Okay, does this make sense? So this is an example how cells intrinsically have the ability to recognize where they are in the cell cycle, and even though they have the capacity in terms of all the proteins that are required to advance to the next phase of the cell cycle, when there are problems, i.e. trouble finishing replication in this case, they choose not to progress through the cell cycle, and that's due to an active regulatory system.
One of the things we exactly is about the relationship between the start of mitosis and CBC2.
Mm-hmm.
What aspect of this would CBC2?
We will very soon get to that.
Oh, good.
Yeah, we're going to talk about that in a moment. Okay, another question? Yeah.
I was just wondering in general about hybrid cells, how can you make sure that the two nuclei don't fuse? I mean, is it like something you do in the procedure? Or?
That the two nuclei don't fuse? Yeah, so the membranes surrounding the cell itself and those that surround the nucleus are very different. And the way that the fusion studies are done is generally treating cells with a detergent that allows fusion of cellular membranes but doesn't allow fusion of nuclear membranes. And that's seen very clearly under the microscope. You see one big cytoplasm and two nicely intact nuclei.
But I mean, in other settings, could you have the nuclei fused? Are there?
There are sort of genetic tricks in yeast that you can use to do that, I think, but I'm not familiar with ways of fusing nuclei in living mammalian cells.
So in that lower cell, that cell went into mitosis even though its DNA hadn't finished.
That's right. Yeah. So that's the point of this, is that normally if you block replication with this drug, the cell will just sit in S phase forever because it knows that it shouldn't enter mitosis because it hasn't finished replicating. However, tricking it with caffeine allows entrance into mitosis even though it really shouldn't be doing that. Okay? And so these types of mammalian cell experiments, cell biology experiments, really sort of stimulated a guy named Lee Hartwell, who is a recent recipient of the Nobel Prize for his work in budding yeast, to really address the idea that cells contain active regulatory systems that monitor progression through the cell cycle. And so the best way of doing that, to convince people that you really have identified a bona fide pathway, is to isolate mutants, genetic mutants, that can no longer perform that function, okay? And so that's what Lee Hartwell did, and that experiment is kind of mapped out on this slide, okay? So Lee Hartwell, again, is working in budding yeast, and in this particular experiment, he takes wild-type yeast cells and just lets them grow. And la-di-da, what do you get? A big colony of yeast cells. That's fine. Now, if you take the same wild-type cells and irradiate them to cause DNA damage, then this is not Hartwell's observation, this has been known for a long time, the cells stop dividing. Okay? So the question is this halt to the cell cycle controlled by an independent regulatory system? Or is it a manifestation rather, of not being able to really adequately repair the DNA? And so to distinguish between those two possibilities, he used a mutant in a gene called RAD52. Now, RAD52 is simply required to repair the double-strand break. That's all that it does, okay? And so in a RAD52 mutant, he found that when you irradiated, you got a cell cycle arrest, But the cells never came out of that arrest because they couldn't repair the damage. So they eventually died. Okay. Importantly, he found a different class of mutants also, and that's exemplified here by RAD9. In a RAD9 mutant, you could take cells, irradiate them, and the thing that they did is they kept dividing. Okay, so they had lost the ability to realize that there was damage present and to stop the cell cycle. This was also ultimately a lethal event because if you don't allow time for the repair systems to actually repair the lesions, and you just carry on through the cell cycle anyhow, eventually that's going to kill the cell just due to gross problems in chromosome distribution. Okay. And so the major difference between a bona fide repair mutant and a checkpoint gene is that the repair mutant didn't affect the ability of the cell to arrest the cell cycle, whereas the checkpoint gene did. Okay. And so this was really formal genetic proof then that cells do contain regulatory systems that monitor the status of chromosomes and can react and control the cell cycle in response to problems. And so since then, there have been genetic screens done ad nauseum in yeast and other organisms to identify checkpoint genes. Okay? And again, the basic criteria is a gene that's required to promote cell cycle arrest in response to damage. And a lot of those genes have been named RAD 1 through 5,000 by now because they are sensitive to radiation, and that's the way that the screens were done, initially just looking for genes that were very sensitive to radiation, and then further analyzing whether or not there were cell cycle defects in them. And so basically, long story short, is that a lot of genetic analysis in different organisms had outlined a pathway in yeast where a number of these different RAD genes were responsible for controlling the cell cycle in response to damage. So all of this was known by the late 1980s to early 1990s. But what wasn't clear at all was how all of this genetic analysis in yeast related to human cancer. Okay? And the big breakthrough there came with the identification of the gene that's responsible for this genetic disorder that I had discussed earlier, ataxia telangiectasia. Now this is a disease. People who have germline mutations in a gene called ATM are born with this disorder, and it has a number of different symptoms. There's neuronal degeneration phenotypes, immunodeficiency phenotypes, sufferers are sterile, there's massive genomic instability, and critically there's an extreme predisposition to cancers of many different types, okay? As well as radiation sensitivity. So this disorder had been known for a long time and been studied for a long time, but the molecular biology really allowed a very clear connection between human cancer susceptibility disorders and that yeast genetics I was talking about, when the gene was eventually cloned and found to be homologous to one of these RAD checkpoint genes from yeast that I had discussed earlier, okay? And in fact, the homology was most striking to a gene from fission yeast called RAD3. And so basically what that says is that the genetic analysis in yeast was really outlining a pathway that was very critical, not only in yeast, but also in human cells, for preventing the eventual onset of cancer. Okay? And that really then justified the use of yeast genetics and biochemistry in lower organisms to understand the biochemical function of these genes so that they could eventually be applied in principle to the diagnosis of human health disorders. So let's talk a little bit about ATM then. ATM is now a family of checkpoint proteins. Humans have two versions. They have ATM itself and a closely related protein called ATR. And both fission and budding yeast also have two versions of this protein. These proteins are very big, 300 to 400,000 Daltons. They are protein kinases. And importantly and interestingly, it has been shown in recent years that the kinase activity of these proteins is inhibited by caffeine. And so getting back to this experiment I discussed earlier, this then provides a molecular explanation as to why cells that are blocked in the cell cycle in response to a replication problem can suddenly advance through the cell cycle in response to caffeine, and that's because caffeine is inhibiting the activity of the ATM and the ATR kinases, and the function of these kinases is to prevent cell cycle progression when there are problems. Okay, so everything sort of came together in recent years with the idea that yeast genes that have been found in genetic screens were similar to human genes that cause predisposition to cancer, that were affected by pharmacological agents that had been shown previously to affect this feedback control in the cell cycle. And so a broad outline then of how cells from yeast to human regulate the stability of their genomes is that when DNA damage is present, ATM and ATR are activated as protein kinases. That activation then serves to prevent instability of the genome, okay? So that should be instability. And so the question is how? How do they do this, okay? And we can talk a little bit about one particular example of how ATM and ATR can regulate the cell cycle. As I mentioned, ATM and ATR are protein kinases, and as most of, if not all of you, are very well aware, protein kinases really serve to modulate the behavior of their substrates. And so you have protein X, which in this case is active and doing the thing that it was designed to do, and then along comes a protein kinase and phosphorylates it, and that phosphorylation then promotes a conformational change in protein X, and protein X can now no longer do what it's designed to do. So protein kinases really control the behavior of other proteins. And so what are some of the behaviors that are modulated by ATM and ATR when cells are exposed to DNA damage? Well, we've already discussed at some length the fact that the cell cycle is stopped or delayed, but that's certainly not the only function of these kinases. Another very critical function is that DNA repair enzymes are organized and activated in a way that we really still don't understand, but it's clear that there's a relationship between activation of these kinases and the ability of the sort of workhorses of DNA repair to access the problem and facilitate repair of the lesion. Another important thing that they do when damage is encountered during S phase of the cell cycle is they actually stop further DNA replication. Okay, and this hopefully makes sense because when DNA damage is present, the cell does not want to use that damaged DNA as a template for duplication, and so the idea is that DNA replication should be in a regulated way attenuated until damage has been repaired. And then under very drastic conditions where there's an overload of damage, these kinases can actually direct apoptotic or suicidal programs that cause programmed cell death and serve to then eliminate the cell from the population so that the organism doesn't run the risk of having that cell turn into a renegade and proliferate out of control. And so really then you can see how the response to damage that's controlled by ATM and ATR is very complicated and involves a number of different pathways, but of course is very critical if damage is going to be stopped before it can cause mutations. Question? No. You can turn it on, it can make it disappear, it can do almost anything you can think of.
Can I just ask you on the tumor suppressor genes, the relationship here? Any relation?
So tumor suppressor genes, I think classically defined, are those that limit cell proliferation, okay? And so they're not necessarily checkpoint genes, which are more concerned with guarding the, ensuring that the genome is stable, whereas tumor suppressor genes function to limit how fast cells divide, okay? And I think you'll be hearing more about them in upcoming lectures, so I won't say too much about them. But suffice to say that they're further downstream from what we're talking about now. These genes here are required to make sure that mutations don't accumulate. When they fail, you can get mutations in tumor suppressor genes, and if that's a loss of function mutation, that can be bad because then cells begin to divide faster than they normally should. Okay, so let's go through, I think there was a question earlier on CDC2, and I can give you one nice sort of biochemical example of how ATM and ATR can control the cell cycle. Okay, and so this diagram here kind of summarizes how cells enter mitosis at the biochemical level. The master regulator of entrance into mitosis is a protein kinase called CDC2 complexed with cyclin B. Those of us who work in Xenopus also refer to it as MPF for maturation promoting factor, and the newest terminology is CDK1, but for the purposes of this discussion, we'll just call it CDC2 cyclin B. Okay? So CDC2 cyclin B accumulates when cells are in interphase. Okay? And because it can accumulate during interphase, cells have developed a system that suppresses its activity until all the conditions have been met for entrance into mitosis, okay? Because once CDC2 cyclin B kinase is activated, that's an irreversible event, and cells are committed to mitosis no matter what's already happened in the recent past, okay? And so the transition from the inactive to the active form of CDC2 cyclin B is controlled by phosphorylation. There's a tyrosine marked here on CDC2 that is a substrate for the Wee1 protein kinase. When that tyrosine is phosphorylated, this CDC2 cyclin B kinase is inactive and cells cannot enter mitosis. There is a phosphatase that acts in the reverse direction of the Wee1 kinase that removes that phosphorylation. And so when that phosphorylation is removed, CDC2 is activated, and then the cell is committed to mitosis. So the battle to enter mitosis can be summarized as a tug-of-war, essentially, between Wee1 that wants to keep CDC2 inactive and CDC25, which wants to dephosphorylate CDC2, activate it, and promote entrance into mitosis. Okay? So this sort of biochemical paradigm had been worked out a long time ago. And the question became, how does, or even if, does the checkpoint regulate this activity? And is this a potential sort of mode of interception that the checkpoint can take advantage of to control progression through the cell cycle? And so the question then, is this reaction a target of the damage checkpoint? And so the way the experiment was done is a very simple experiment. It's a very nice experiment. That tyrosine I told you about, you can go in and you can mutate it. You can get rid of it, change it to phenylalanine. And those two amino acids are chemically very similar, so it doesn't affect the kinase activity of CDC2 at all. But it does have a profound effect on the ability of CDC2 to be regulated. Okay, so if you change this tyrosine to a phenylalanine, then CDC2 is no longer subjected to inhibitory phosphorylation. Normally that's okay because, for reasons I won't get into in the interest of time, there are other mechanisms that limit CDC2 activation to just prior to entrance into mitosis. The interesting observation came, though, when cells expressing this mutant were exposed to DNA damage. They entered mitosis regardless of the fact that they had damaged DNA. Okay? So I think this addresses your question here. The way that the checkpoint prevents entrance into mitosis is that it controls the phosphorylation status of this tyrosine. So that if you change that tyrosine to phenylalanine and you eliminate that level of control, then cells are no longer able to arrest the cell cycle in response to damage. Okay? This is actually a G2M thing. This is trying to prevent entrance into mitosis. Similar, roughly similar things are thought to happen in G1, although the data there is not nearly as good. And so essentially what we have then is checkpoint control of the cell cycle acting by controlling phosphorylation of this tyrosine, such that when there's DNA damage present, the CDC25 phosphatase is inhibited. That allows Wee1 to win the tug-of-war and hold CDC2 in the inactive state, and you prevent entrance into mitosis. Okay, and so that general pathway then is summarized here. And this is an example, really a very nice example of how at the biochemical level we have a very complete understanding of how cells respond to DNA damage and control the cell cycle. So the first step is that damage is detected. ATM or ATR kinase is then activated. That activated kinase directly phosphorylates a second group of kinases called CHEK1 or CHEK2. Those kinases then become activated. They target CDC25. They directly phosphorylate CDC25. That has the opposite effect on CDC25 in that it turns it off. Okay, so phosphorylation from here to here results in attenuated CDC25. That allows CDC2 to remain in the phosphorylated form, and consequently cells don't enter mitosis. Okay, so this is a very good example of how in just the past 10 years we've been able to go from yeast genetic analysis, which is does the colony grow or not, to a real biochemical pathway that describes how checkpoint pathways do what they do and why it's obviously important to have intact checkpoints if you want to be able to deal with DNA damage in a reasonable way. And so in the last couple of minutes then, I'll just turn to the general topic of how cells actually detect the damage in the first place. Okay, because this is really, as far as this particular pathway goes, this is certainly the most poorly understood event. We know that you have a cell with its chromosomes. The evil scientist treats those cells with DNA damaging agents. Voila, we have activation of these kinases, but we don't really understand how the kinase can see the damage, can see that it's different than normal DNA, and therefore activate itself and go to do its thing. And so the last part then will be the beginnings of a description of the molecular mechanisms that allow cells to detect DNA damage. And so as I've mentioned a few times now, there are two general categories of checkpoint kinases in human cells. One is ATM and the other is ATR. And the reason that apparently that there are two is that they respond to very different types of DNA damage. So for example, ATR concerns itself mostly with sort of small chemical modification of DNA. Okay, and the best example of that is what happens when you get a sunburn. When cells are exposed to ultraviolet light, that causes thymine dimers or UV photoproducts in DNA, which is a chemical modification of the base within the DNA that makes that DNA a very poor template for replication, as well as causing problems for transcription and other things. ATR recognizes damage that's caused by other types of mutagens, such as alkylating agents, which simply methylate DNA, or oxidizing agents. And this is actually a very common cause of DNA damage. Any organism that has to breathe in order to live, in other words, uses aerobic conditions, generates free oxygen radicals as a byproduct of respiration. Occasionally, the scavenger systems that make sure that those free oxygen radicals don't do bad things to us don't quite work, and those free radicals can oxidize DNA and damage it. Okay, and so ATR then responds to that. So ATR is concerned with a broad class of lesions, but all sort of chemically modified bases. In contrast, ATM really only cares about one thing, and that's when the chromosome is broken into pieces. Okay, and that's otherwise known as a double-strand break. And so a lot of what I've already told you is basically the use of ionizing radiation as an experimental means to produce double-strand breaks in DNA. So ATM can somehow tell that this piece of DNA has been cut in two, okay, and it is then activated. And so now, in the last couple of minutes, I'd just like to present some primary data that gives you guys an example of how we go about figuring out how cells respond to damage and some of the basic sort of parameters of that recognition event. Okay, so the next few slides are all work from a guy named Mike Kasten, who's at St. Jude's Hospital in Memphis. And he's been interested in how the ATM kinase is activated. Okay? And what they found is one of the very first things that occurs that you can detect in cells when you hit them with DNA damage in the form of ionizing radiation is that the ATM kinase phosphorylates itself, okay? And that's called autophosphorylation. Now, whether or not the same molecule can kind of like bend around and phosphorylate itself, or two different guys come together, is not clear, but nonetheless, ATM autophosphorylates in response to DNA damage. And so because that seemed to be a very important, very early event in the whole damage recognition pathway, one thing that became important was to figure out where within the ATM kinase that phosphorylation event occurred. Okay? And so the way that people do that is kind of diagrammed here. You take cells and you feed them radioactive phosphate so that when a protein of interest is phosphorylated, it becomes radioactive. You then hit cells with ionizing radiation. Every time you see this GY, it refers to gray, which is just a unit of radiation, okay? And so this is then inducing DNA damage, basically. And as you can see, when you induce damage, you see a big difference in the amount of radioactivity that is incorporated into ATM. What we're looking at here is ATM. And so the idea then is to figure out where within the ATM kinase this phosphorylation event is occurring. The way that that's done is the protein, the radioactive protein, is isolated out of the gel away from the rest of the guys, digested with a protease that cuts it into little tiny peptides. Those peptides are then run out on TLC plates in 2 dimensions to separate them, okay? The only peptides that show up in this experiment are those that are radioactive. And so the major peptide that's radioactive is this spot here. There are zillions of different peptides all over this plate, but this is the major radioactive one. So this peptide is then excised from the gel and sequenced, okay? And so to make a long story short, what they were able to sort out is within the ATM sequence, it's this particular serine here that gets autophosphorylated in response to DNA damage, okay? And so that then allowed them to make a very useful reagent. And that's the whole point of introducing this, which is that they were able to use this peptide, and in particular the phosphorylated form of this peptide, to generate an antibody that only recognizes the phosphorylated form of ATM. Okay? Does everyone understand that? So when you use this as your antigen to make an antibody, because you've put the phosphate on the serine, the antibodies that come out of that immunization will only recognize phosphorylated ATM. And that's shown very nicely here in a Western blot, where cells were either hit with DNA damage in this lane or not, and then probed with this phosphospecific antibody. And as you can see here, a band of the size of ATM is only noticeable in response to damage. Okay, so now they have a marker where they can go in and look at activation of ATM in response to damage, because this autophosphorylation only occurs when damage is present. And so that allowed them to do a series of what I think are really very interesting and very nice experiments, just describing the basic parameters of how ATM is activated. So in this particular experiment, they're interested in the amount of time it takes for cells to mount a response to damage, okay? And so the way they do that is they take a dish of cells and they damage them with, in this case, half a gray of irradiation. And then they make lysates from those cells and probe with that phosphospecific antibody. And as you can see here, even in the 0 time point, they're generating some cross-reactivity. And the next time point is 5 minutes after damage, and they're seeing full activation of ATM. And so the bottom line is, as fast as they can hit the cells with damage, get them out of the dish, and lyse them to make the lysates, they're already seeing activation. So this is a very, very rapid response, probably less than a minute to get it going in the first place. The other thing having this antibody allow them to do is get a feel for how many double-strand breaks within a particular genome are required to mount an ATM response. And so to do that, instead of adjusting the time, they kept the time constant in this experiment and adjusted the amount of damage. Okay, so now you're getting increasing amounts of DNA damage. And we know from older studies how many double-strand breaks are produced in response to a given unit of DNA damage. And what we find here is that as few as 0.1 grays, which is only 4 double-strand breaks throughout the entire genome, is sufficient to activate a detectable level of ATM. So again, this is a very impressive response, because you got to remember the genome is very big. And to be able to detect just 4 double-strand breaks amongst all of that DNA within the nucleus in a short amount of time is pretty impressive. And so that's what was found there. And so the next question then is an interesting one, and that is how much ATM is activated in response to a set number of double-strand breaks. So in this experiment, they decided that they were going to introduce 18 double-strand breaks. And then they wanted to see how much of the total ATM within a given cell was activated in response to those 18 double-strand breaks. And so the way this experiment was performed is that cells were damaged, then lysates were prepared, and immunoprecipitated with that phosphospecific antibody. Okay? So this immunoprecipitation reaction then is going to capture all of the activated ATM out of these cell lysates. Then they did a second immunoprecipitation, and they just scooped up what was left after this first immunoprecipitation. And for that, they used antibodies that recognize ATM regardless of its phosphorylation status. So in the first IP, you're going to pick out only the activated form of the molecule, and in the second IP, you're essentially going to pull out whatever's left. Okay? And so then the question is, can you figure out how much was in the first IP relative to the second IP? And those results are shown here. This is the amounts by Western blot of material that was collected in the first immunoprecipitation. And as you can see, it's more than what was collected in the second precipitation. Okay? This band is bigger than this band. And one can do more quantification, but this picture alone reveals the fact that the amount of ATM that was activated in response to just 18 double-strand breaks is more than 50% of the total pool. Okay? And that's pretty impressive again, because calculations have shown that there are millions of ATM molecules per cell. So when you have 18 double-strand breaks, you're activating millions of ATMs, and that obviously implies a very robust signal amplification mechanism that's occurring in the detection response also. And so then just to summarize some of these parameters of ATM activation, we know that double-strand breaks are the main trigger for ATM. That ATM autophosphorylation is really the first detectable response. Interestingly, very low levels of double-strand breaks are sufficient to activate most of the ATM in the cell, okay, implying again signal amplification, which, as you can imagine, would be important for mounting a robust response to relatively low amounts of damage. And, but that still leaves open the question of exactly how ATM is recognizing the double-strand break. And I do have some stuff prepared for that. But I think in the interest of time, since we're out of it, I'll just skip that and go to the end here, the summary. And basically, this slide kind of summarizes what we know about how cells respond to double-strand breaks. They can occur through a number of ways. In the lab, it's typically by ionizing radiation. That activates ATM. That's a very rapid response and a very sensitive response. And then the activated ATM can then go and arrest the cell cycle, as well as promote DNA repair and stabilize other sensitive intermediates. And the net result is that the cell is allowed time to repair this damage before it continues on through the cell cycle. Okay? People who lack ATM and therefore the ability to really process and guard against double-strand breaks, those people are viable. They live. I mean, they have horrible lives, but this is not an activity that's essential for the viability of the cell itself. Now, the other guy I talked about, ATR, if you take ATR out of a cell, that cell dies. And so if you use that as a criteria, then one might argue that cells can actually deal with double-strand breaks better than other forms of damage. However, eventually it's going to cause major problems. There is a relationship clearly between the length of telomeres and how that length is controlled and decreased over time, and the ATM pathway, which actually might serve to protect telomeres that have shortened beyond the point of being healthy, might protect cells from the deleterious effects of that. So telomere shortening can cause chromosome fusion. And what is known is the ability of defects in telomere length to promote chromosome fusion is enhanced dramatically in cells that lack ATM. Okay. But again, the molecular nuts and bolts are still not in place. But there's an obvious connection between ATM and the maintenance of telomeres, certainly. No, those are probably separate effects of caffeine. Yeah, I should say the amount of caffeine that is required to see these effects on ATM is 5 millimolar, and any pharmacologist will tell you that that's absurd and much too much to be a very specific inhibitor. I think caffeine's effects on cyclic AMP turnover occur at far less concentrations, far lower concentrations. So it's probably more specific.